Hi, I'm Hunter Scott, and today we're going to be talking about development of B cells that interact with vaccines to give us immunity, and this is sober biochemistry. Most people think that the process of building immunity starts the moment that you go into the doctor or CVS or wherever and say, I want a flu shot. And pop, there you go. Good job. You're all done. Congratulations for being an adult. And it really begins long before the vaccine is even administered in the bone marrow, where a hematopoietic stem cell has given rise to a young lymphocyte progenitor, or a pre-lymphocyte. After she gets some stimulation from her morning fix, she's ready to take on the world. When I grow up, I want to be a lymphocyte. I'm ready keeping the body a better place and fighting off evil, blah, blah, blah. Now, this future lymphocyte contains about 100 variable or V gene segments, several diversity D and joining J gene segments in the loci for its antigen receptor. These genes are inherited from its parents, and basically all of these proteins are the same until VDJ recombinase comes in and he's like, hey, I think you'd be a great B cell. Let me look it up. Look at you. You'd be great. I don't know. Can I be a T cell or a B cell? No. This is what you're going to be. You're going to be great at it. Okay. And the VDJ recombinase recombines these gene segments on the future lymphocyte. The different combinations of variable diversity and joining segments, as well as changes in the nucleotide sequences at the junctions of these segments, make the immunoglobulin receptor of this future B cell completely genetically unique and specific to one antigen. VDJ recombinase is the product of recombination activating genes 1 and 2, or RAG1 and 2, which make double-stranded nicks in the lymphocyte's DNA to cut out intervening gene sequences until there is a region with adjacent V, D, and J segments, the signature sequence for B cells. The RAGs require a divalent metal ion to be able to make these cleavages. Before the lymphocyte can graduate to a mature B cell, it has to go through some testing and checkpoints to make sure that it can do its job right. If the VDJ recombination wasn't productive, then the pre-B cell can't produce the proper receptors that all mature, naive B cells have, immunoglobulin M and immunoglobulin D. And it's like, sorry, see you later. You just didn't make the cut. And that's what negative selection is. But if the recombination was, say, a little too successful, the immature B cell can go rogue and just think everything's an antigen. It's kind of like, bad? No. Bad? No. Bad? No. Bad? No. Bad? That's enough. But if this somatic recombination yielded antigen receptors that bind to antigen with just the right affinity, the lymphocyte can pack its bags and head off to the lymph node armed with immunoglobulin M and D class receptors. It is now locked and loaded. off and ready. Lethal weapon for microbes. Okay, so wait, let's back up here a second. I mentioned immunoglobulin M and immunoglobulin D. The term immunoglobulin is interchangeable with the term antibodies. Antibodies can either be membrane-bound receptors on B cells that bind to antigens that activate the B cells. And also B cells can secrete antibodies that mark them for phagocytosis or neutralize the antibodies the antigens or the microbes. Vaccines also contain primarily weakened or disabled microbes, bacteria, or viruses. Basically, the things that don't cause infection, but rather the pieces of the things that do. So that way the B cells, enough of them, so that way the B cells can recognize it for future interactions with or exposures to these pathogens. So our B cell is hard at work in the follicles of the peripheral lymphoid organs, hanging out, ready to get activated when a macrophage comes in with antigen from the vaccine and is like, "Hey, I found this outside. I don't know if you want it." It better not be gross. Oh my god, this is what I've been looking for. My life is solved. And then the T helper cell comes in and is like, "Let's do this." And then they go nuts, and antigen receptors begin their work on receptors inside the cell. So inside the cell, there are membrane-bound receptor proteins that form the B cell receptor complex along with the immunoglobulin. And they have cytoplasmic tails who have tyrosine residues that are in a conserved motif that are phosphorylated when the B cell receptors recognize the antigen from the vaccine. These phosphotyrosines recruit an additional kinase, SYK tyrosine kinase, that phosphorylates tyrosine residues on adapter proteins that activates them, beginning the signaling cascade for activation of the B cell, which includes proliferating through clonal expansion, secreting antibodies, and recruiting other lymphocytes to the site of infection, among other things. And in the goal of the vaccine, remain in the body as memory B cells long after the infection is cleared. To produce memory B cells, the lymphocytes undergo high-affinity maturation. As the lymphocytes rapidly proliferate in the germinal center of the lymphoid follicle, the genes of the clones undergo numerous point mutations. Then these clones are tested for their ability to bind antigen on antigen-presenting cells and present that antigen to follicular T helper cells. If they can't do that, it's like, what the heck? You had one job! Ugh! Of the ones that pass high-affinity maturation, a small fraction of these high-affinity B cells end up not secreting any antibodies at all, but become memory cells instead. They patrol the body and are ready to respond rapidly if the antigen from the vaccine is reintroduced, providing potentially a lifetime of immunity. Some of the antibody-secreting B cells, or plasma cells, migrate to the bone marrow, where they can survive even years secreting antibodies. It's kind of dark and scary in there for an antibody shop, but you know what they say, location, location, location. Most vaccines, like the flu vaccine, stimulate the production of neutralizing antibodies that B cells secrete to block infection from spreading initially. These antibodies coat microbes and prevent them from being able to bind to and enter host cells. The end.